Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this lecture, I will be going over digestion of dietary proteins and absorption of amino acids. Now let's get into the details. Digestion of dietary proteins, it begins in the stomach and is completed in the small intestine. Now the enzymes that participate in the digestion of dietary proteins are produced in their inactive form called zymogens. And the zymogens need to be activated in the small intestine to their active form. And how that is done? So the proteolytic cleavage of zymogen into their active form is necessary for these zymogens to be active in the small intestine, thereby they can participate in digestion of dietary proteins. Now note that these enzymes, that is protein digestive enzymes, they are stored in their secretory granules in the 𝗓𝐽𝐽crine part of the pancreas in their zymogen forms. The reason for that is, if they are there in the active form, they would have digested the pancreatic proteins itself. That's why these enzymes, they need to be there in their inactive form called zymogen forms, and only when they come to small intestine and when there is food there, so only during that time they need to be activated, and then they participate in the digestion of dietary proteins. Once the digestion is done, once the action of the enzyme is done, these enzymes are digested among each other. Basically, they kill each other, means they digest one another, and that's how their action will end. Otherwise, if they continue to function in the small intestine, then they will digest even the small intestinal proteins also. Okay, that's the kind of information we should have before we begin. So, zymogens are activated only when they are necessary. And another point that you need to remember here in protein digestion is, no single enzyme completely digests a dietary protein. They all need to have a concerted action. The reason for that is, so let's get into see what is the specificity of each one of the enzymes that are participating in the digestion of dietary proteins. Now the digestion of dietary proteins in the stomach. Now as you know, our gastric parietal cells, they secrete hydrochloric acid, and the chief cells, they secrete pepsinogen. Now the pepsinogen, it is a zymogen. It is secreted in its inactive form and it has to be converted into its active form called pepsin, and that is done in the presence of hydrochloric acid HCl. And this type of activation of pepsinogen into pepsin without any enzyme being participating in this proteolytic cleavage and activation process, that type of activation we call it as autocatalytic mechanism. Now once this pepsin is active and the pepsin is going to break a peptide bond in a protein molecule, the active pepsin, it is going to act on a denatured protein that is there in the stomach according to its specificity. Before we get into the specific action of pepsin, let's see what actually happens to a dietary protein when it reaches stomach. Dietary proteins are denatured by the presence of acid in the stomach, and the denaturation, as you know, it is the loss of secondary, tertiary and the quaternary structure. Because of this denaturation process, when a non-covalent interactions are removed and protein secondary, tertiary, quaternary structures are opened up, and a polypeptide chain is in an open form with only primary structure remaining, which is holding one amino acid with another amino acid only with the peptide bond. Now this particular protein is ready for digestion. And the digestion of this polypeptide chain with individual amino acids attached with the peptide bond, it will be acted by a pepsin enzyme in the stomach. Now note that pepsin is an endopeptidase, meaning it is going to act on the peptide bond within the polypeptide chain. As you can see here, I have written a small polypeptide here just to show you how the action goes on. So on the left side you have amino terminus, on the right side you have carboxy terminus, and some of the amino acids are linked by the peptide bond here. I have not written a specific amino acid, I just wrote a amino acid general. Now endopeptidases are the ones that will act within the polypeptide chain and break the peptide bond. So that means they are not going to act on the amino terminus peptide bond, that is the number 1 peptide bond, or they are not going to act on the carboxy terminus peptide bond, that's the last peptide bond. Other than the first and the last peptide bond, endopeptidase can act on any other peptide bond. That's the meaning of endopeptidase. So pepsin is an endopeptidase, so it will act within the polypeptide chain. So I will just take an example, and it is going to act on here where I am highlighting. Okay, it's going to break that peptide bond. Now what is the specificity for breaking that? In order to explain you the specificity there, I need to write down specific amino acids in a polypeptide chain. So here it is. So here I have written a short polypeptide with specific amino acids, and there are 4 peptide bonds that I have written. So one start from the methionine and go all the way to cysteine. So the methionine on the amino terminus side and cysteine on the carboxy terminus side, and 4 peptide bonds with methionine making a peptide bond with the glycine. So look at the atoms that are participating in the peptide bond formation. It is the carboxyl side of methionine making an interaction with the amino group of glycine. That's how you should read the peptide bond there. Okay. So in number 1, as I am saying here, it is the carboxyl side of methionine making a peptide bond with the amino group of glycine. Like that, carboxyl group of glycine making a peptide bond with the amino group of arginine. Like that, as I move on, carboxyl group of arginine making a peptide bond with the amino group of tyrosine, and the carboxyl group of tyrosine making a peptide bond with the amino group of cysteine. Why I'm just repeating this? It's because these protein digestive enzymes, like the one that I am trying to explain here, that pepsin, which is an endopeptidase, it is going to act on the carboxyl side of aromatic and acidic amino acids. Okay, so the specificity of pepsin is, you got to remember the specificity, it is the carboxyl side of aromatic and acidic amino acids. That means in order to understand protein digestion and absorption, you must have finished amino acid structure, and you must know classification of amino acids based on their side chain characteristics, and you must know what is the meaning of, means what are all acidic amino acids, what are all hydrophobic amino acids, what are all basic amino acids, and sulfur containing amino, all that classification you should know. Okay, with that understanding, pepsin is very specific for the carboxyl side of acidic and aromatic amino acid. Now look for acidic and aromatic amino acid in the list. So methionine is not acidic or aromatic, glycine is not acidic or aromatic, arginine is not acidic or aromatic, tyrosine is aromatic amino acid, and then cysteine, and it ends there. Now, does the pepsin is going to act on this polypeptide chain? Note that I told you the very first peptide bond and the very last peptide bond in a polypeptide chain cannot be broken down by endopeptidase. That's the first thing, because pepsin is an endopeptidase. So where is the bond that is made by the tyrosine? So the bond that is made by the tyrosine that you have found here, there is tyrosine which is aromatic, which fits into this. Pepsin is specific for carboxyl side of acidic and aromatic amino acids. So tyrosine is there, which is aromatic. So according to that, we are selecting aromatic tyrosine. Now look at where exactly the peptide bond made by the tyrosine. Tyrosine makes a peptide bond with the cysteine, which is the last amino acid. That means it's a last peptide bond. So that peptide bond is not cleaved by endopeptidase, because that should be done by the 𝐒𝐧𝐼. 𝐒𝐧𝐼, they will take care of the last peptide bond in a polypeptide chain. So there is a amino terminus 𝐒𝐧𝐼, N terminus amino peptidases, and C terminus carboxypeptidases. They will take care of the first and the last peptide bond. Anything in between will be taken care by endopeptidase. So here in this example that I have purposely written, that there is no place for pepsin here in this sequence to act on it, because the one that is available is the last peptide bond. It cannot be taken care by the pepsin now. So how to understand the concept? What I will do now is I am going to increase one more peptide bond so that you understand exactly what it is. So I am going to add one more amino acid to explain the concept. So we have CONH, and then I will write lysine, which ends with COO minus. That's the carboxyl side of lysine. So we have one more peptide bond here. Okay. Now the carboxyl side of cysteine is going to interact with the amino side of amino group of lysine. Okay, now let's see. Now your peptide bond, that is number 1, 2, 3, 4th peptide bond, where tyrosine is contributing the carboxyl group to amino group of cysteine, and now that is an endopeptidase can act on this, because it's no longer it's a last peptide bond. There is another last peptide bond is there. So endopeptidases can act on this. Now pepsin, since based on its specificity, the specificity is carboxyl side of aromatic and acidic amino acid. Where is the aromatic amino acid here? Tyrosine is an aromatic amino. Where is the carboxyl side of tyrosine? Left side is amino group, right side is the carboxyl group. Always we write it like that. So right side of the tyrosine is carboxyl group, which is contributing to the peptide bond, right? So that's where, that is the peptide bond it breaks. So the peptide bond that is broken now is this peptide bond breaks, because it is the carboxyl side of tyrosine. Carboxyl side of tyrosine is the right side of the tyrosine. That's how you need to understand. Very important. This is what you need to understand the protein digestion. Carboxyl side of tyrosine is broken up. That peptide bond breaks and the things are separated. That means there will be release of these two amino acids as cysteine and lysine separated, and rest of the molecule will be released, will be methionine, glycine, arginine and tyrosine separated. Like this, a long polypeptide chain is broken down according to the availability of the peptide bond based on the specificity. And that is what is the action of pepsin on polypeptide chain, which has opened up because of the acidity and undergoing a partial digestion process. Okay, so that is how a pepsin works in the stomach. So the figure here is showing you parietal cells are the ones that will release hydrochloric acid. Chief cells are the ones that release pepsinogen, and pepsinogen is converted to pepsin by the presence of hydrochloric acid, and once pepsin is active, it is going to break proteins, peptides and small amino acids. Now the thing is, pepsin has got a specificity, and we understood that in the previous slide. So the specificity of pepsin, which you must remember, is pepsin acts on the carboxyl side of acidic and aromatic amino acids. Now let's look at digestion of proteins by enzymes coming from the pancreas. So the protein digestion in the small intestine. So the partially digested proteins in the stomach, mediated by the action of pepsin, comes into the duodenum as the acidic content enters into the duodenum after the gastric contraction. So this acidic content, it is going to release cholecystokinin and secretin, the endocrine hormones released from the duodenum, and cholecystokinin and secretin will go to pancreas and bring pancreatic juice, which contains pancreatic enzyme, and secretin releases bicarbonate into the pancreatic juice, and the bicarbonate, it is going to neutralize the acidic content that came from the stomach, and the pancreatic enzymes are participating in the digestion process. But only thing that you need to remember here is these pancreatic proteases. Protease means the enzyme that digest proteins are referred as protease, which is a general term. So pancreatic proteases, they are secreted in an inactive zymogen form, just like what we have seen with pepsinogen. And these inactive zymogens, that is pancreatic zymogens, they need to be activated first, and that activation, it will be done by a brush border located enzyme called enteropeptidase, which is also called as enterokinase. What this enterokinase does is, it is going to activate first trypsinogen into trypsin. Okay, so one of the pancreatic zymogen that is released by the exocrine pancreas is trypsinogen. So the trypsinogen that is released from the pancreas need to be activated into trypsin, and that is done by enterokinase enzyme. And this enterokinase, it is an active enzyme which is located over the brush border epithelium, and as soon as trypsinogen arrives into the intestine from the pancreas, zymogen is activated into trypsin. And once trypsin is active, this trypsin is going to convert other zymogens, that is chymotrypsinogen into chymotrypsin, and it's going to convert pro-elastase into elastase and pro-carboxypeptidase into carboxypeptidase. All that is done by trypsin. So active trypsin is going to take care of activation of other zymogens, and also trypsin, further it is going to convert other trypsinogen into trypsin itself. So it all takes an activation of few trypsinogen into trypsin, so that trypsin can take care of activation of all other zymogens. That means if there is any mutation in a gene that is coding for enterokinase enzyme over the brush border epithelium, that will lead to no activation of any zymogens or decreased activation of zymogens, because enterokinase is the one that will initially activate trypsinogen into trypsin, then trypsin is activating all other. So mutation or a defective enterokinase will lead to protein malabsorption, because that is the one which initially activates trypsinogen into trypsin. And if there is a mutation in pepsinogen molecule which is not activated into trypsin, that can also lead to protein malabsorption. Okay, so I have written here, active trypsin catalyzes the conversion of chymotrypsinogen into chymotrypsin, proelastase into elastase, and procarboxypeptidase into carboxypeptidase. Now we have active enzymes. They are trypsin, chymotrypsin, elastase. All these are serine proteases that are endopeptidases. They are also endopeptidases. Now, what is the specificity of these activated enzymes, that is trypsin, chymotrypsin, elastase? Specificity is, you need to remember, trypsin is going to act on carboxyl side of lysine and arginine. Trypsin is going to act on carboxyl side of lysine and arginine. I have already explained you what is the meaning of that carboxyl side. And the chymotrypsin is going to act on carboxyl side of hydrophobic amino acids. And elastases will work on the carboxyl side of small side chain amino acids like glycine, alanine, like that. So this is the specificity of trypsin, chymotrypsin and elastase. Based on those specificities, these enzymes will work and break a polypeptide chain into smaller fragments. Now we have something called exopeptidases. So we have two exopeptidases, that is procarboxypeptidase A and procarboxypeptidase B. Now these procarboxypeptidase A and procarboxypeptidase B, both of them are activated into their carboxypeptidase form by again trypsin. Now the action of carboxypeptidase A is on the carboxy terminus hydrophobic amino acids. That means if you look at the right last peptide bond, that is the carboxy terminus, the last peptide bond in a polypeptide chain or in a peptide, a last peptide bond, if it is contributed by hydrophobic amino acid, all you can look at is, if the last amino acid is a hydrophobic amino acid, then the last peptide bond is clipped by carboxypeptidase A. If the last amino acid is lysine or arginine, it is clipped by carboxypeptidase B. So this is what is the specificity of carboxypeptidases, which are exopeptidases. It is there in the name carboxy. They are working on carboxy terminus amino acid, last amino acid in the towards the right side, carboxy terminus. Now, with the action of all these enzymes, that is pepsin, trypsin, chymotrypsin, elastase, carboxypeptidase A, carboxypeptidase B, with their specificities, the polypeptide chain is broken down all the way almost into dipeptides, tripeptides and individual amino acids. So what will happen to the small peptides, oligopeptides, di and tripeptides, which are still there in the lumen of the intestine after the digestion of enzymes that came from pancreatic, repeat. So what will happen to the small peptides, oligopeptides, dipeptides, tripeptides that are released from the digestion of a polypeptide by the action of pepsin, trypsin, chymotrypsin, elastase, carboxypeptidase A and carboxypeptidase B. So the further digestion of these small oligopeptides will happen over a brush border epithelium located aminopeptidases. Aminopeptidases, they will cleave one amino acid at a time starting from the amino terminus. It is an exopeptidase. It's going to remove one amino acid at a time by breaking the very first peptide bond on the amino terminus. Very first peptide bond is the amino terminus peptide bond. So, and then individual amino acids are absorbed, including di and tripeptides are also absorbed. And on di and tripeptides, they are digested into individual amino acids inside the cytoplasm, and that will be done by intracellular peptidases. So like this, with the concerted action of all these enzymes, a long polypeptide chain is broken down into individual amino acids which are ready for the transport. What all the enzymes we have? So let me write down the list of enzymes that we have seen. One is starting from the stomach, we have pepsin, then we have trypsin, then we have chymotrypsin, we have elastase, we have carboxypeptidase. In that carboxypeptidase A, and then we have carboxypeptidase B, and then we have aminopeptidase, and then we have intracellular peptidases. So all these enzymes are participating in the digestion of a polypeptide chain, and they have their own specificity. Just to revise that, pepsin is carboxyl side of pepsin, pepsin acts on carboxyl side of aromatic and acidic amino acids. Trypsin is going to act on the carboxyl side of arginine and lysine. Chymotrypsin acts on the carboxyl side of hydrophobic amino acids. Elastase acts on the carboxyl side of small group side chain amino acids like glycine and alanine. Carboxypeptidase acts on the carboxy terminus, the last peptide bond of hydrophobic amino acid. Carboxypeptidase B act on the carboxy terminus, last peptide bond contributed by lysine and arginine. Aminopeptidases act on the first peptide bond, the amino terminus. There is no specificity, it removes one at a time starting from the amino terminus, especially on the small peptides. And intracellular peptidases, they are in the cytoplasm, they work on di and tripeptides without much specificity. So like this concerted action of all these proteolytic enzymes produced by stomach, pancreas and intestine, they digest dietary proteins into individual amino acids. That is what is shown here in the figure. Trypsin is there in the center here because trypsin is the one that is going to activate all the pancreatic proteases once it is activated by enterokinase. First enterokinase has to activate trypsin, and once trypsin is active, it is going to activate all other zymogens. So with all that, so protein is digested into individual amino acids, and these amino acids are ready for transport into the portal blood. So here is the table that summarizes all the things that we have seen so far. So it shows activation of pepsinogen into pepsin, and the activation is autocatalytic mechanism, and it's an endopeptidase and act on aromatic and acidic amino acids. Trypsinogen into trypsin done by enterokinase, it's an endopeptidase and thus acts on carboxyl side of lysine and arginine. Like that, all other zymogen activation and the information is given here, and you need to remember all these points, very important. So some of them are endopeptidases and some of them are exopeptidases. Now coming to absorption and transport of amino acids. Now the amino acid absorption is through secondary active sodium dependent co-transport mechanism. Now the di and tripeptide absorption is also by secondary active proton dependent co-transport mechanism, and the sodium gradient, it will be created in the enterocytes by sodium potassium ATPase, and this helps in the absorption of other dietary macromolecules like carbohydrates, simple form glucose, galactose, fructose also. Intracellular peptidases convert dipeptides and tripeptides into amino acids, and amino acids undergo facilitated diffusion into the portal blood. And that is what is shown here in the figure. So, amino acid absorption through sodium gradient, and the sodium gradient is maintained by sodium potassium ATPase. Di and tripeptide absorption is through the proton that is getting into the intestinal cell. As it is getting in, so di and tripeptides are absorbed. Di and tripeptides are digested by intracellular peptidases and amino acids are released from it, and whenever amino acids are concentration is increasing, so whenever there is increase in the concentration of amino acids inside the enterocytes, so they will be getting out into the portal circulation by facilitated transport mechanism. Now in the absorption mechanism, there is a role of a glutathione which is participating in the absorption of amino acids across the intestinal lumen into the enterocyte, and that is explained by a gamma glutamyl cycle which is also called as Meister cycle. So it runs in the intestine and also in the renal tubular cells where reabsorption of amino acids are going on. So glutathione, it carry amino acids inside the cell, and in this process glutathione is resynthesized. The major role of gamma glutamyl cycle is in fact is the synthesis of glutathione, because many tissues lack transpeptidase and 5-oxoprolinase activities which are necessary for the synthesis of glutathione. As you see here in the figure, here is the gamma glutamyl cycle called as Meister cycle, where glutathione on the cytoplasmic side, it will be attached with a amino acid, and that is done by gamma glutamyl transpeptidase enzyme. During this process, a cysteine glycine is separated and amino acid is attached with the glutamyl group, so you get glutamyl amino acid, cysteine glycine is separated, and this cysteine glycine further it breaks into cysteine and glycine, while glutamyl amino acid is releasing its amino acid inside the cytoplasm and glutamate is separated. Now the glutamate, cysteine and glycine, all of them are brought together and glutathione is resynthesized. That means in the gamma glutamyl cycle, when the glutathione is taking this amino acid in and further itself broken down and it is reformed. So this is one way of synthesizing glutathione in a cell. Now transport of intact breast milk antibodies in the newborn. The concept here is the mother's milk contains antibodies, and those antibodies should not be digested, because these are the antibodies that are secreted by the mother for the fetus to use those antibodies, so that these antibodies can protect them until they develop the immune mechanism. So the breast milk contains antibodies released by lactating mammary glands. Out of all immunoglobulins, IgA is the one that coats the intestinal surface to protect mucosal cells, and these antibodies are absorbed intact in newborns and neonates, because newborn has relatively low acidity, thereby immunoglobulins are not denatured, and also greater intestinal permeability that is seen in newborn will allow absorption of intact antibodies, and those antibodies will protect a newborn against infections until a newborn develops his or her own immune system, so that they can protect against the invading organisms. Alright, now let's move on to see clinical aspects of protein digestion and absorption of amino acids. Our very first disease which is related to this concept is cystic fibrosis. In cystic fibrosis, there will be a mutation in a gene that is coding for CFTR protein, that is cystic fibrosis transmembrane conductance regulator, where there will be decrease in chloride secretion into the duct. Because of that, So the fluid media that has to be present in the duct to carry exocrine pancreatic secretions, so that doesn't go on. So that means pancreatic digestive enzymes, they don't come into the duodenum. Because of this, what will happen? So the pancreatic digestive enzymes don't flow into the duodenum and they don't participate in the digestion of macromolecules that are coming from the diet. That is, there will be decrease in the digestion of carbohydrates, proteins and lipids. That's why in cystic fibrosis, there will be malabsorption of all three macromolecules, that is proteins, carbohydrates and lipid malabsorption. Coming to our next disease, that is heart-nerve disease. So in the heart-nerve disease, there is an autosomal recessive condition where there will be mutation in a gene that is coding for a transporter that is participating in the absorption of tryptophan along with other large neutral amino acids in the intestine and also reabsorption of these in the renal tubular cells. Because of this, what will happen? So the specific amino acids including tryptophan are not reabsorbed and also not absorbed from the intestine, overall leading to deficiency. One of the most deficient amino acid in heart-nerve disease is tryptophan, and the deficiency of tryptophan will lead to niacin deficiency and that eventually lead to pellagra signs and symptoms. Now cystinuria is caused by defect in heteromeric amino acid transporters referred as HATs. This is due to mutations in SLC3A1, SLC7A9 and also SLC7A7 genes. So this heteromeric amino acid transporters are coded by SLC3A1, SLC7A9 and also SLC7A7. So these are the genes that are mutated that will lead to defective heteromeric amino acid transporters. These defects will prevent a proper reabsorption of basic or positively charged amino acids along with cysteine and ornithine. So under normal circumstances, this protein allows certain amino acids including cysteine to be reabsorbed into the blood from filtered fluid that will become urine. Mutation in either of these genes disrupt the ability of this transporter protein to reabsorb these amino acids, allowing them to become concentrated in the urine. So as the levels of cysteine in the urine increases, the cysteine crystallizes. So these cysteine crystals, they will have a hexagonal shape when observed under light microscope. And this is what is seen in cystinuria, lysiniuric protein intolerance. Now let's move on to see our next disorder is protein losing enteropathy. So in this particular disease, so patients will show loss of proteins in the feces because some of these proteins are not absorbed because of the defect in the intestinal mucosal cells. Now coming to acute necrotizing pancreatitis, this is an inflammation and ischemia of pancreatic tissue which leads to acinar cell damage in the exocrine pancreas, and that will lead to abnormal activation of trypsin inside the acinar cells. Once the trypsin is activated, it's going to, as you know, it will activate all other zymogens into their active form and they start digesting a pancreatic tissue. And pancreatic autodigestion, it is in the central event in the pathogenesis of acute necrotizing pancreatitis. Now the methionine malabsorption syndrome, it is an autosomal recessive disorder where there is a malabsorption of methionine, and then the methionine in the intestinal lumen, it will be converted into alpha hydroxybutyrate by the bacteria. And because of this alpha hydroxybutyrate, which is a degradation product, so let me write it down, it is alpha hydroxybutyrate, which is the digested product of methionine by the bacteria, and this will have a characteristic smell of dried malt. So the smell of dried malt is seen here, which is referred as outhouse. That's why it is called as outhouse urine disease. And the dried malt smell is because of alpha hydroxybutyric. Make sure you remember that. And patient may show mental retardation, seizures, white hair, diarrhea, and generalized edema. So these are some of the clinical aspects related with protein digestion and amino acid absorption. So I hope in this lecture you have understood how our dietary proteins are digested by concerted action of various enzymes, and also how amino acids are absorbed, and we went through some of the clinical disorders that are related to protein digestion and absorption of amino acids. Thank you.